Write your name and roll number in the chat box. Please write your name and roll number in the chat box. Good morning everybody. So last class we can discuss about the protein drug delivery system, how to deliver the protein as a drugs. But there is some problems we discussed elaborately. There is a physical and chemical stability problem in case of the protein drug delivery system. But still nowadays it was established. So many drugs already available in the market. Some drugs are in the pipeline and some drugs on the research going on research. So how to formulate the protein as a drug delivery system? Before that, how to isolate and purification of the protein? Suppose X is the protein which is a good therapeutic activity, but how to isolate or how to culture it, where it will be cultured and where it will be collected from it? It was a human, animal, plant, bacteria or microorganisms. Generally the protein is cultured in the bacteria because the bacteria is the protein in your uh, insulin, the recombinant DNA technology. This protein is a insulin is nothing but a proteins. This protein is isolated from the bacteria, E. coli. So how to isolation and what are the purification methodology can use for the use of proteins or therapeutic proteins? So today we discuss about how to isolate the proteins from the different source and what are the purification methods of the proteins. And after that it will be formulated as a drug. Let us go. And what are the objectives of this study? That describe the most common method of protein isolation and the purification. What are the most common methodology and simplest methodology which are can used the isolation, that means separation of protein from the plant, animal or the bacteria or from the other sources. And simultaneously what are the purification process, because we are getting the animal or plant, it is a raw material, but it will be purified. After that it is 100% pure and it will be utilized for the formulation of the drugs. Next, number 2, the compare between the different method of protein purification method. There is a comparison statement which is CPR, which is the most easier, and how to get the more accurate or more pure form of the proteins. There is a comparative statements. Number 3, construct a purification and algorithm based on of your knowledge in the protein purification process. So we are getting some knowledge and it will be algorithm or it will be emphasized on your protein purification technology. So these are the basic concept before to start the purification of protein, isolation and the purification. Next, so basically we are collect the protein or isolate the protein from the bacteria or the microorganism, because we know the bacteria contain the 30s ribosomes and this ribosome is the factory house for the protein synthesis. So in case of bacteria, generally broadly it will be classified into two groups. One is the gram positive, another is the gram negative. So you can utilize this molecular and cell biology, this gram positive and gram negative, how to form or synthesize a protein, and that protein which are utilized by the bacteria, as well as sometimes this protein having some therapeutic activity, and how to isolate the protein from the bacteria. So before that we can just thorough about this structure of the bacterium, because in the last class we discussed about last semester discussed about the molecular cell biology, what is the structure of the bacteria and how to synthesis of the protein inside the bacteria. Now what are the steps to isolate or separate the protein from the bacteria, then it will be purified. So just you can see what are the structure of the bacteria. It is the gram positive and there is the gram negative bacteria, because this gram negative bacteria and gram positive only difference, it is the this cell wall is composed by the peptidoglycans and the cell wall is composed by the basically the lipid contents. So the mesosomes and the peptidoglycans, cytoplasm and flagellums, these are the gram negative in case of the flagellums, periplasm, ribosome. So basically the ribosome or the 30S or 50S, this ribosome, which is the main factory house for the protein synthesis. After the protein synthesis, how it will be isolated and what are the steps involved, the isolation, or what are the different methodology can use the separation of proteins, then how it will be purified. So basically the extracellular and intracellular, some proteins which are synthesized, basically protein are synthesized in the intracellular inside the cell in case of ribosome, but some it will be released from the inner cell to outer cell. So these are the process by which the protein will be released. This is by the exocytosis process, the release of the protein from inside to outside, or it is endocytosis, that means the protein released from outside to inside, that will be released. So these are the extracellular, these are the two types of protein. One is the extracellular protein, another is the intracellular protein, and that protein can be isolated or separated by the different method from the bacteria. Isolation of extracellular protein, no need of cell disruption. Isolation of extracellular protein and isolation of intracellular protein. If it is intracellular protein, then need of the cell disruption, because if you not disrupt the cell, that protein will not surface out. But in case of extracellular cell, that means outside the cell, there is no need of cell lysis or cell disruption, because just outside the cell you can just separate it. But in case of intracellular cell, it must be cell disrupt, otherwise the protein cannot be separated out. Or number 2, secreted, the soluble protein can be collected in the cell supernatant after the configuration, from the centrifugation. In case of extracellular protein, there is no cell disruption, no need of cell disruption. Only you can the suitable solvent used, that will solubilize the protein and centrifuge the protein molecules, that will be solubilized into, then it will be separated. Another is the membrane bound protein might be released from the cell simply using the detergent. Some proteins which are extracellular but it is bound in the membrane, in that type of protein, that will be separated by simply using some detergents, because this protein adhere with the membrane molecules, and the detergent which can use this detergent just like a used as a emulsifying agent to reduce the interfacial tension between the membrane and the protein molecules, and the protein molecule will be free, then it will be centrifuge, or just like a second methodology, or that means some solvent are used to solubilize the protein and it will be centrifuge out. So isolation of extracellular protein, only you can use the 3 steps, there is no need of cells, only 2 steps, there is no cell disruption is required rather than the intracellular protein, and it will be solubilized with the particular solvent, and a supernatant liquid will be collected after the centrifugation process, and the membrane bound protein might be released from the simply using the detergent. If it is membrane bound, the simple detergent can use it, that will be used it to separate the protein from the bounding of the or adhere with the membrane. Next, isolation of intracellular protein, need of cell disruption. These are the very easiest method comparatively than the intracellular method. But in case of the protein which is present inside the cell, so how it will be separated it? First, need of cell disruption. Obviously, the cell or protein which is present inside the cell, if you do not disrupt this cell, how it will be collected? Okay. So number 1, what is the disruption methodology? First one is the detergent lysis. The detergent which can use it into the cell, and here is just one tube moves slow down, and the pastel run motor and Teflon plastic, where the cell tissue sucrose homogenate in the tissue. It is rotated in the RPM 600. Number one, detergent can lysis. So detergent is the one of the most important that cell can be lysis. Then the another methodology which can be used, the enzymatic lysis. That means where the enzymes used for the cell for lysis or breakdown of the drugs. Another is the osmotic lysis, because in that case there is a change of the osmolarity of the outside and inside of the cell, and that must be disrupted. That means if the change in the osmolarity, if it is inside it is the higher the osmolarity, then automatically it will be disrupted. Another is the freeze throw cycle. There is another methodology, another ultrasonication procedure by which you can disrupt the cell wall, and another is the homogenizer. Homogenizer, that means all the cell will be extracted, then material will be homogenized, a particular all the component will be surface out, then the particular protein can be isolated by the different methodology, especially for the cell column chromatography or liquid chromatography. So any question regarding this up to this? What are the difference between the extracellular protein isolation and the intracellular protein isolation? They are extracellular, there is no need of cell disruption, and the very simple method by which you can separate the protein. Achha, this topic not only the theory but also that is in case of the practical record, practical style, it will be provided in the next class. But today we discuss about based on the theoretically how it will be allocated, and next you go to the time we are discussing about the practical oriented, the how to say lysis by the detergent or by the enzymatic lysis. But in case of protein will be disrupt, in case of the intracellular protein that will be by the different methodology detergent. Do you understand up to this? Hello.
Yes, sir.
Okay, so in case of intracellular there is so many methodology you can apply for only the isolation. Okay, next, this is the isolation of intracellular protein. This isolation of intracellular protein, how it will be isolate. First, the first step of the purification is the most protein in the disrupt tissue, and the cell is controlled by the control fashions. Number one, using the gentle mechanical producer that called the homogenization. You can know it, the homogenizer, which is the machine which is extract the cell, and this plasma membrane of the cell can be ruptured so that the cell content of the release, and the four commonly used procedure that are shown in the figure. There are 1, 2, 3, 4. These are the procedure. The first one is the cell suspension or tissue. It will be homogenized first, then the mechanical stirrer you can use. There's 4 steps, 4 procedure or methodology we can use to separate it. First, the break the cell with the high frequency sound, that means ultrasonic sounds. That high frequency sound, that means break the cell. Number 2, use the mild detergent to, suppose this protein will be separated, but 2, this use the mild detergent to make a hole into the plasma membrane. That means the mild detergent to make a hole, because this hole for help for the protein release from inside to outside. And number 3, force cell through this small hole using the high pressure. Number 2, this pore, a high pressure created, that will be the intracellular protein which is surface out through hole. And number 4, a shear cell between the closed filter rotating plumber and a thick wall for the glass vessels. So these are the 4 technology you can apply for after the isolation of the intercellular material for the cell suspension or the tissue. Either break the ultrasonic sound or using the some detergent and force, and the holes will be, and the force will be applied, the intercellular protein which is released out and surface out and outside the cell. And next, step is the when the carefully applied the homogenization leaves from of the membrane bound organelles intact. These suppose these are the two proteins. This protein which is extract after this process will be over, this protein will become, then it will be separated. This is the isolation, then it will be purified. Next the purification. Do you understand these slides? How to isolate the intracellular protein. Suppose this protein which is inside the cell, how it will be separated? Obviously the cell lysis and different cell lysis methodology can discuss with you. And next the 4 steps of reaction or steps of the different 4 methodology you can apply it to separation of or isolation of protein. Do you understand? Any question regarding these slides? And also on paper also supplied, you can details about this, how to separation of protein. Do you understand? Next steps, the purification of the protein. After the collection or separation of protein, that protein is the raw protein, which are not used as a therapeutic activity, which are not used for the drug deliveries or used as a drug. Before that, even some any chemicals which are synthesized in the factory, that will be not utilized for the formulation. That will be before the utilization, that will must be purified at least near about to 100%. So in that case, the protein, that means it will be collected from the bacteria or by the extracellular or from the intracellular. That protein contains some others impurities. That will be rejected or that will be separated or that will be purified by the different methodology. First, what are the methodology you can apply it for the purification process? Number 1, differential centrifugation method. Differential centrifugation, you know it, we can separate. In case of blood, we are knowing the centrifugation proceed, the higher molecular protein due to the centrifugal force that will be settled down in the test tube. This is the centrifuge process. But here you can use the differential centrifuge method. That means one centrifuge cannot be separate the protein, and step by step so many centrifugals are procedure that can separate the that can separate the protein. That's why it is called the differential centrifugation. That means one centrifugation is not enough for the purification. So many centrifugation process step by step, ultimately to collect the 100% pure form of protein. That is called the differential centrifugation. Number 2, differential salt precipitation. Suppose this homogenate which are precipitated with the different types of salt, and that types of salt can be removed after it will be bind with this particular salt, and the salt will be removed, you can get the absolute proteins. Number 3, differential solvent precipitation. We know the different solvent having the different solubility of the different protein. So which preparation or which protein which are soluble in the which solvent, that will be first determined first, or you can use arbitrary methodology which this preparation or the protein which are solubilized with the solvent, and that solvent which is separated out with along with the proteins, and that will be separated by the HPLC or column chromatography methodology. Another is the preparative electrophoresis. In case of electrophoresis, that means preparative, any other SDS or page, any type of electrophoresis, because protein are the high positive charge and negative charge, they are will be due to the high electrophoretic method that can be separated by very easily. Another is the column chromatography, and I think it is the most cheapest and easiest method by which you can separate or purify the protein from the homogenate or homogenizer. So these are the 5 methodology we can apply it for the purification of the proteins. Do you understand? Hello,
Yes, sir.
বুঝতে পেরেছো? or sometimes more than one
হ্যাঁ স্যার
approach, যে একটাই procedure, there is a single procedure
হ্যাঁ
is not enough. Sometimes how the differential centrifugation after their column chromatography, or differential solved out then chromatography. So more than one approach may be required. So that means one procedure cannot to separate out the or purify the particular protein. Sometimes two or more procedure can applied to purify the protein. So first one is the differential centrifugation motion. This is the centrifuge in how it will be. First, it will be homogenized into the mixer grinders. There will be the bottle cells. It contain the subcellular content component. That will be this will be contained in the tubes, and the centrifuge separated in the 800 into 800 into the gravity in the 10 minutes. তার মানে 800 rpm, the gravity is for 10 minutes ধরে it will be rotated in the 10 minutes. Then the nucleic will be sedimented. Then another centrifuge, that's why it is called the differential centrifuge. The centrifuge approach 50,000 rpm into gravity 10 minutes. Then the mitochondria or lysozyme, and these are the sediment it will be. The first one is the nucleic acid with the 800 rpm, and that's 15,000. There the mitochondria or lysozyme or another material will be sedimented. Then from this extract another centrifuge into the 1 lakh rpm, that will be the fragments of flagella membranes and the endoplasmic reticulum will be settled down. So next steps with the centrifuge is the 2000 rpm, and the centimeters of the cytosol or the ribosomal sediment will become, because this is our main product, because ribosomal sediment is contain the protein. That will be this portion, it will be separated by the column chromatography. So 1 2 3 4 5, so 5 steps, 5 different centrifugal procedure that can separate or purify it, separate as well as the purification of a particular protein by the differential centrifugation process. Only this is centrifugation means only one process. When the blood biochemistry practical, the only one procedure, the one centrifuge tube, you take the blood sample into the cubet, then the centrifuge 5 to 10 minutes for 5 to 4 to 5,000 rpm, the serum will be separated. That means the cell rbc or wbc others, it will be settled down. But this is called the centrifugal. So it is called the differential. Why it is called? Because the differential centrifugal procedure can ultimately collect the ribosomal sediment, because this ribosomal subsequence sediment is main essential component for the protein that will be separated or purified by the column chromatography. So first is the differential staining, because the 5 differential centrifugal process, after that it will be collect the ribosomal sediment, and that will be purified by the column chromatography. Do you understand what is the differential centrifugates? Understand? Or any problem, please ask me freely. Hello?
হ্যাঁ স্যার বোঝা গেছে।
বুঝতে পেরেছো?
হ্যাঁ স্যার বোঝা গেছে।
বোঝা গেছে। তার মানে একটা আরপিএম এ দেখো ডিফারেন্ট আরপিএম, আমরা প্রথম 8,800, দেন 15,000, দেন 1 লাখ, 2 লাখ, এই যে ডিফারেন্ট মেথডোলজি, ডিফারেন্ট সেন্ট্রিফিউগাল, সেই জন্য বলা হচ্ছে ডিফারেন্সিয়াল সেন্ট্রিফিউগেশন। এন্ড আফটার দ্যাট উই collecting the ribosomal sediment. Next, the differential centrifugation, separation of protein or any other material on the basis of their size, mass and density. This is not a standard, which is basically depend upon the mass density of the protein, and it is the function of size of the protein that will speed the centrifugation. Obviously the speed is the one of the most major important factors to centrifuge them or separate them, because the centrifugal force which is separate out the protein or higher molecule or separate the different types of molecular substances, মানে different molecular weight substance, and it is gives us rough separation. It is basically fractionating, fractionation করা হচ্ছে, it is rough, but after the column chromatography it will be separated out. Next, another is the dialysis, differential dialysis method. In case of, you can know, I have a term, in case of the dialysis, when the blood is not purified, or is some toxic material or a drug or poison, in that case the drug, or in case of creatinine level high, in that case you are use some dialysis technology. But here also this homogenate separated a protein by the dialysis technique. What is the start? At start by the dialysis at equilibrium condition. Suppose these are the dialysis bag, dialysis membrane, which are contain the homogenate, and it will be placed into the suitable solvent and the concentrated solution. It is the concentrator because these dialysis bag contain the proteins and others molecule which are more concentrated and which are placed into the particular solvent, and that particular solvent which are dissolve the particular proteins. So after that, after few hours, that means this the red molecule, which is the desired molecule, or it is having some therapeutic activity, which are dissolved in that particular solvent, and after a few hours this protein will be dissolved out, and finally it will come out. Hmm. Hmm. পার্টগুলো আচ্ছা, হ্যাঁ হ্যাঁ, অ্যাব্রিভিয়েশন ল্যাটিন, ওগুলো তো বলতে পারছি না। ও শিডিউল, শিডিউল, ও এক্স আর এক্স আর এক্স, এক্স আর ইয়ার, আচ্ছা ঠিক আছে, তারপরে হুম, ও তো বলতে পেরেছি, ওগুলো তো পেরেছো, হ্যাঁ। হুম হুম হুম, 70 বললে কেন, কোন অসুবিধা নেই, তারপর বলো। না, সেটা কমাবে কিন্তু যদি কেটে দেয়, যে লোক গেছে বলছো না, তার তারপর কি বলছে, তারপর ওরা কি বলে? হুম, আর জিজ্ঞেস করেনি এত, বাবা তা তো রিক্স হয়ে গেল। তো বুঝতে পেরেছি, কিন্তু জিজ্ঞেস করে যে কে পাঠিয়েছে। হুম হুম, হ্যাঁ, হুম, ঠিক করে। রিহারটা বলেছে, কেসটা দেখছি। ঠিক আছে, আমি এবার দেখে নেবো পরে। হুম, hello, sorry, do you understand the dialysis? Hello, Ronabo, hello,
হ্যাঁ
hello,
স্যার।
dialysis টা
হ্যাঁ স্যার।
বুঝতে পেরেছো? Just it is a dialysis bag which contain the homogenate just placed into the solvent. The after the sometimes due to the osmolarity the protein will be
হ্যাঁ,
come out. ঠিক আছে?
কাম-
বেসিক দ্য ডায়ালিসিস, যে মানে বডিতেও আমাদের ডায়ালিসিস করে থাকে, ধরো টক্সিক ম্যাটেরিয়াল, পয়জন, snake bite, in that case dialysis will be done the based on the osmotic pressure, and it is allowed to get the rid of the most salt ion, and it is impossible to remove the salt completely. এটা তার মানে this is the process, there is a no মানে pure 100% substance cannot be get, Next, the column chromatography, the most common and best approach. This is the most common and best approach to purify the larger amount of the protein by this column chromatography. Do you know the column chromatography? You can know what is the chromatography, basic principle of the column chromatography, and achieve the highest level of the purity and highest amount. That means the column chromatography and nowadays the HPLC, high performance liquid chromatography, which is achieve the highest level and purity and largest amount of the maximum amount of the drug or protein can be isolated or purified by this column chromatography and requires low effort. Actually, low effort, that means column chromatography one of the technique. Just you can apply the homogeneous, automatically it will be separated out as a band, and the lowest likelihood to damage the protein product. Obviously, this is another most advantageous method because there is no heat can be applied in this methodology. So all types of proteins, heat labile to heat stable, all types of protein can be separated by the column chromatography, because in case of column chromatography and standard method of pharmaceutical industry, there is these are the main methodology. Even there some drugs will be extract by the column chromatography. Do you any question from this column chromatography? I think it will be discussed in your pharmaceutical analysis what is column chromatography, thin layer chromatography, and different types of chromatography based on the affinity, based on the adsorption, based on the others things. Do you know the column chromatography? Hello?
হ্যাঁ স্যার কলাম ক্রোমাটোগ্রাফি করেছি
হয়েছিল তো?
হ্যাঁ
That will be discussed in the earlier class in the pharmaceutical analysis, I think.
হ্যাঁ স্যার
সেই জন্য
অ্যানালিসিস
I do বলো।
স্যার
হ্যালো?
হ্যাঁ স্যার কলাম ক্রোমাটোগ্রাফি তো স্যার ভাই এর সেপারেশনের জন্যই করেছিলাম এতে
মানে ও
অ্যানালিসিস
তো ইউজিতেও ছিল, পিজিতেও ছিল, কিন্তু প্র্যাকটিক্যালি হাউ ইট উইল বি অ্যাপ্লাইড, কি প্র্যাকটিক্যাল হয়েছিল?
হ্যাঁ স্যার প্র্যাকটিক্যাল হয়েছিল এটা করে
কলাম কমান্ডার প্র্যাকটিক্যাল হয়েছিল না এইচপিএলসি হয়েছিল?
না স্যার এইচপিএলসি হয়েছিল
কলাম ক্রোমাটোগ্রাফি না হলে ইউটিউবে আমি দেখাচ্ছি যে কলাম ক্রোমাটোগ্রাফি, যদি এখন এইটা ক্লাসটা নিয়ে নেই, ঠিক আছে? কলাম ক্রোমাটোগ্রাফি ইজ দ্য মোস্ট ইজিয়েস্ট মেথড, জাস্ট লাইক এ দেখতে অনেকটাই হচ্ছে পিপেডের মতো, সরি পিপেড বলছি মানে বুলেটের মতো।
বুরেটের স্যার
বুলেটের মধ্যে তার মধ্যে কিছু অ্যাডজর্বেন্ট দিয়ে দিলে সিলিকা জেল, এবারে ডিফারেন্ট পোলারিটি অনুযায়ী দ্য সলভেন্ট উইল বি প্লেসড or homogenized will be placed above the column, then the due to the affinity or adsorbent or different principle, that will be formation of a different types of band, and it will be separate or collect out on the different color, and it will be protein that will be detected by the different protein detection method or protein coloring agents. Okay, so in case of three methodology, one is the gel filtration, ion exchange, and the affinity. Gel filtration, ion exchange, and the affinity, these are the three methodology can applied for it for the column chromatography. These are the gel are used, and after that the first is the porous material which is poured into the column, then the homogeneate or that will be separated out, that will be placed over the porous material, then due to their affinity, that will the first test tube nil, second test tube nil, third and fourth which is contain the protein molecules or desired components. I think clear? This is called the gel filtration. Yes. Next
হ্যাঁ
is
স্যার
the next, যদি কোনো প্রবলেম থাকে বলো, যে এইগুলোই প্র্যাকটিক্যালি দেখাবো আমি, মানে this is the topic which is in the practical as well as the theory. I know it how to separation. Next is the, and this is the very good technique, even advanced technology can applied, even a very useful technique nowadays, how to separate the protein on the different basis. Next is the ion exchange. Here based on the gel filtration, here it is based on the ion exchange, because protein are the positive and negative charge, and ion also positive and negative charge. So cation and anion, there is some reaction, that will be the same methodology, same types of procedure, just here only gel, here is the ion exchanger is used. So cation and anionic exchange, then the solution poured into it, and ultimately it will be sort of 3 to 4 test tube which is contain the this therapeutic proteins. Next affinity, that means adsorbent. There is some silica gel which are used for this adsorbents, and due to some affinity there is some produce some the band, or these are the compound which is produced first initially those compounds 6 and 7 test tube. So these are the affinity, based on the affinity some compound which is polar and some compound which is non-polar, due to the solvent polarity it will be formation or separation separated out by the due to their affinity. So in case of column chromatography, based on the three mechanism, one is the gel filtration, number two is the second ion exchange, and number three is the affinity. You can you can apply it for this which is best suitable for this protein, or you can apply it initially. There suppose this protein is unknown, how initially gel filtration, if it is not separated in the ion exchange, then you can use the affinity chromatography. And I think this affinity chromatography which is the most generalized method for separation of protein. Next, the gel filtration, aka size exclusive or the molecular exclusion chromatography. Molecular the separated according to the difference in the size and by the through the hydrophilic polymers. And in case of gel filtration, this polymer bead composed of the cross-linked dextran or dextrose which is highly uniform porous like Swiss cheese, porous like substances, gel filtration chromatography, that will be that means these are the substances and these are the porous like substances, only this particle can pour into it and it will be separated out, and larger protein come out first and smaller protein come out last. Especially because in the gravitational force, obviously the large molecular weights of substances can be separated out in case of gel filtration rather than the lighter substance or small protein. Next is gel filtration technology, and you can apply for the know-how or the protein, the molecular size makers and adds a target to get proteins. As for example, you do not know purify enzymes and separate to the tips, and you will see it will be appears. Okay, the porous polymer beads or protein mixture are added to column chromatography by cross linkage, and proteins molecule are separated by the size. Larger molecule passes through the freely and appearing in the carrier function, or earlier in case of larger molecule than the smaller molecule. Next is the ion exchange chromatography. It is based on the principle to separate the basic on the adsorption and highest resolving power and highest loading capacity and widespread capability. Most frequently chromatographic technique or protein purifier, this column chromatographication. So these are the ion exchange, there are positive and negative charge. In that case, then protein also positive and negative charge, which is proteinic substances which is used in which chromatography. Next is the ion exchange chromatography. Ion exchange, this ion exchange process based on the concentration of a counter ion, and these represent the ionic exchanger. These are the procedure by which the 3/4 column which is positive or negative charge. Next is affinity or adsorption separation, in which the molecular purification methodology and reverse bindings or adsorption to a chromatography. In case of thin layer chromatography, same in the based on the adsorption only. Binding substance ligand immobilized to the insoluble porous molecule, and purification 100x and better for the single steps and highest, and prepare for the method of if possible. Next is affinity chromatography. These are also general protein of the column. Just as per the affinity of this particular chromatography, it will be separated out. Next is the isoelectric focusing, because the isoelectric pH, at that pH there are positive and negative compound. The same at the low pH or in the low pH, that means in the acidic pH, low pH in the acidic pH, the high pH minus charge isoelectric focusing, that means in that case a certain pH where the positive charge and the negative charge in equal in case of the proteins. In case of low pH, and this is the high pH, here the positive low pH positive charge, there are acidic substance positive, here is the negative positive negative, and ultimately this type of band can become in case of low pH and in case of high pH. Another is the polyacrylate gel or gel electrophoresis. You know, in case of gel electrophoresis, there is a positive cation and anionic substances which is placed over the gel, and the gel is applied either polyacrylate gel PEG or in the SDS gel, sodium rhodesyl sulfate gel is used, and this placed over the homogenate or the particular proteins that will be placed, and then run the gels, and ultimately it will be collected in the cation or in the anion. This is the mixture of macromolecular substance, direction of the electrophoresis, and after electrophoresis देखो, in case of there is a separation in the yellow, green and brown color. These are the three types of molecule, mixture molecule, which is separated after electrophoresis due to upon the cationic and ionic charge or electrophoretic separation. This is the most suitable method, and that will be graphically represented into this. This is the largest and this is the smallest. These are the largest band, that means higher amount. This is the lower amount. So this can, and in case of the preparative electrophoresis, this is the 2D polyacrylate gel or 2D gel or SDS page gel. This in the gel electrophoresis you can use this SDS or the page. So these are the molecular size of this pH. Depend upon the molecular size, it will be separated out on based on the gel electrophoresis. That describe the most common method of protein. So thank you very much for listening the lectures. Or this is not only theory, but next start the practical class also. The how to separation of protein by the column chromatography or by the gel electrophoresis, any other methodology we can apply, or by basically the centrifuge. Just the protein and protein just started your journey. Thank you very much for listening a lecture. If any question, please ask me or in the question in the chat or my chat box. Okay, thank you once again. Or yeah, hello, hello.
হ্যাঁ স্যার।
কোনো কোশ্চেন থাকলে বলো। You any
না স্যার, ঠিক
question?
আছে। কলাম ক্রোমাটোগ্রাফি বোঝা গেছে স্যার।
Column chromatography তো বোঝা গেছে, কিন্তু but before that why column chromatography is used? Basically our main target is the separation of protein, then purification.
হ্যাঁ স্যার।
Separation different methodology can used it, and then there is a different procedure which are used for the purification procedure, and how it will be applied. It practically is very important and very interesting to separate the protein and purification of protein. Next we start a practical up to কত বাজছে 12টা বাজতে যায়. So column chromatography you can see it, or a direct isolation purification of protein. Just you can apply it.